They have Jasmine Foo from the University of Minnesota, Twin Cities, and she'll be talking about site frequency spectra for neutrally evolving tumors.
Hi, everyone. First, I'd like to thank the organizers for organizing this workshop and the opportunity to talk to you about this work. And as mentioned, I'm going to be talking about site frequency spectra of neutrally evolving tumors. This is joint work with Einar Gunnarsson and Kevin Leader. And I would say the work was mostly driven by my student, Einar Gunnarsson. Yeah, so the goal of today's talk will be basically to tell you all about how we can leverage some analyses of branching process models to interpret tumor genomic data. So it'll be a pretty applied talk. And I also will get into some of the limitations and challenges of looking at real data. Okay, so cancer typically initiates and progresses via the accumulation of genetic mutations. And here is an example of the mutations that are involved in the initiation of head and neck squamous cell carcinoma, which is a type of cancer that affects the linings of the oral cavity. So you might start out with an initial cell that's maybe undergoing some kind of clonal expansion, and one of its progeny picks up a mutation that leads to another clonal expansion, and so on. And biologists are very interested in identifying the mutations that occur during cancer progression, and also figuring out what they do, since that might give us some clues about how to treat it. So in the biology community, there's a lot of interest in right now in identifying the so-called modes of tumor evolution, or some details of the evolutionary strategies of tumors. And so this is a figure from a biomedical journal. You can see a few of the sort of hypothesized modes of tumor evolution in the biology community. In one regime, we can think of each mutation sort of arriving, so a different color represents a different mutation, arriving and being sufficiently fit to start a clonal expansion that then takes over the tumor before the next, and then subsequently another mutation arises, and so on. And on the other hand, you can think of a regime where mutations are effectively neutral or have little effect on fitness, and maybe you have many overlapping mutations that don't take over the tumor. Then you have intermediate models, of course. So I'm not really defining these modes mathematically here, but just kind of illustrating the kinds of questions that people in the cancer biology community are thinking about. So one of the specific questions of interest right now is in determining whether or not tumors have evolved under neutral evolutionary processes, and also whether or not we can determine sort of the quantitative parameters of the evolutionary processes governing tumor growth. And I'll discuss what I mean by, what quantitative parameters in a few slides. So in particular, there's been a lot of work done in using tumor genomic sequencing data, and specifically this thing called the site frequency spectrum to try to answer these questions. And before I kind of get into that, I will just note that the site frequency spectrum has been used for a long time to develop tests of neutral evolution in fixed size population models, such as the Moran or Wright-Fisher model that Jason introduced the other day. And in this talk, I'll be considering the site frequency spectrum in more of an expanding population under branching process model. And we'll talk a little bit about how these things relate to each other. So to describe the site frequency spectrum, suppose we have a cancer phylogeny shown here. And usually we have a tumor of monoclonal or single cell origin. And let's say our original cell divides and one of its progeny dies and another one picks up a mutation A. And then subsequently it divides and one of its progeny dies, and then the other one picks up another mutation, B. So now this cell has two mutations, and this process goes on and on. And at the end, when the tumor is sequenced, it looks like this in the last row. And the site frequency spectrum is constructed from this single point sequencing analysis of the sample, and it records the number of mutations that are found at any given frequency in the tumor. So here there are, so this would be the site frequency spectrum. There are 4 mutations, E, G, H, and I, that are found in exactly one of these cells, 2 mutations, C and F, that are found in exactly two of the cells, and so on. And then as a note, just on terminology, we use the term clonal mutations to refer to mutations that are found in every cell in the tumor. And so typically you would think of those being the mutations that have accumulated prior to the tumor growth in the initiating cell. But of course, we can still have an alternative route to clonal mutations, as depicted here, where you can have a mutation. The initiating cell has a mutated daughter cell whose sibling dies, so that every cell in the resulting site frequency spectrum has mutation A. You can see the site frequency spectrum, or the SFS distribution, leaves out a lot of useful information about the evolutionary history. It's a pretty crude summary statistic, but it still has been found to be informative about cancer evolution, and it's very easy to calculate from genomic data, which is why it's used a lot today. Okay, so I'll next introduce the population dynamic model that we're going to use to model the tumor growth that leads to this kind of data. Let Z0 represent the size of the tumor population at time t, and typically Z0 is 1. And cells divide at rate R0 and die at rate D0 in a standard branching process model. Each time a cell divides, we're going to have a random number of neutral mutations accumulate in each daughter cell with mean W. So we assume only that this random number of mutations is non-negative and integer valued with a finite mean W. And just some notation, omega infinity is the event of non-extinction, as defined here, and then P0 is the probability of extinction given by D0 over R0, and Q0 is the complementary survival probability. So in terms of prior results, in the context of expanding tumor populations, there has been some prior work on understanding the shape of the site frequency spectrum under this neutral evolution model. And in particular, there have been some approximations derived by considering only the skeleton of the process, which is the process where you consider only the individuals that have an infinite line of descent. So in particular, if we let F be between 0 and 1, representing a frequency, and we let M sub F be the number of mutations found at a particular frequency greater than or equal to F at time t, then Durrett in 2013 showed that as t goes to infinity, then the expected value of this quantity is approximately W over Q0 times 1 over F minus 1, where, as you remember, W is the average number of mutations accumulated in each daughter cell after division, and Q0 is the survival probability of the process. So what we'll see later is that this approximation only holds at the large frequency end of the site frequency spectrum, so mutations that show up in a large number of cells. It was also derived in some other works later on, and it's a cumulative version of a related result, which is that if Sj of t is the number of mutations found in exactly j cells, j bigger than or equal to 1 at time t, and n is the tumor size at time t, then as t goes to infinity, the expected value of this quantity is approximately given by this expression here, which sort of gives the name the 1 over j squared law or the 1 over f law. Okay, so in terms of application, Williams and colleagues in 2016 suggested a neutrality test for tumors, tumor evolution, based on a goodness of fit between the large frequency mutations of the SFS and the 1 over j squared law. And this same set of authors, along with some other groups, used the result to estimate W over Q naught, which is the effective mutation rate, which turns out to be an effective mutation rate on the skeleton process. So as you can see from the form of the result, though, it's not possible to separately identify W and Q naught using this law. So that's one of the things that we're interested in doing. And this is a figure from this Williams et al. Paper. So you can see the actual SFS of a colorectal tumor sample, just to give you some idea of the limitations involved in looking at real data and comparing to sort of these theoretical results. So one thing to note here is the x-axis is the allelic frequency, taking into account the fact that humans are diploid. So the numbers here represent half the percentage of cells in which the mutations are observed. So the authors in this paper chose to interpret any mutation that shows up at allelic frequency 0.25 or more as being clonal. And so allelic frequency 0.25 or more means that the mutation shows up in half or more cells of the sample. And so they're interpreting those as mutations that show up in every cell in the sample. And they cite sample impurity and sequencing noise as reasons for this assumption. And then at the same time, they consider the SFS only in the regions above 0.12 allelic frequency, citing detection limitations as well. So in practice, for their neutrality test, they consider the shape of the SFS in the region from 0.12 to 0.25 allelic frequency, and then compared this to the 1 over j squared law. And they performed an R-squared goodness of fit test, and the results are here. These are different kind of sets of colorectal cancer samples. So they set the threshold of the R-squared test at 0.98 to be considered neutral. And then they found that, in this set of tumors, 4 out of 7 were neutral, 31 out of 82 were neutral, and so on. So this just gives you an idea of how this tests are applied. And I'll just mention that this paper has been somewhat controversial in the community, with subsequent authors pointing out some insufficiencies or limitations that call into question the reliability of these neutrality calls. For example, Bozich et al. Of this result here calculated the probability that under a selection model, you would be able to observe a non-neutral mutation within this exact frequency range, 0.12 to 0.25, and found that it would actually be quite small. And then another group, Day et al., showed that with the experimental and sequencing noise typically present in these kinds of data, a tumor undergoing selection might still have a high probability of giving rise to an SFS that has a very good fit to the 1 over j squared law. Okay, so as I mentioned, the prior work on this SFS is based on approximations, which consider only the skeleton of the process. And as we'll see, that applies only to the large frequency end. And then, so we're interested in obtaining a more complete understanding of the SFS, both at small and large frequencies, and some of our results suggest a future way forward for estimating W and P naught separately using the small frequency end of the spectrum. So we start again with the same notations. So for SJFT is the number of mutations that are found in J cells at time t, and we condition on the event of survival until time Tn. Tn is a fixed time in this set of results, which is at which the population has expected size n, conditioned on surviving until that time. And here lambda is the net growth rate, which is greater than 0. And I'll just mention that this fixed time spectrum is in particular relevant for lab experiments where you are able to know the exact age of the tumor. So at this fixed time, we can calculate the expected value of the spectrum using standard techniques. Condition and non-extinction is given by this expression here, which as n goes to infinity looks like this. And note that when p naught is 0, so there's no cell death, then this is going to match up with the prior 1/j² law that we saw in the previous slide. But one thing that's interesting is that the two parameters W and P naught decouple in this full spectrum where we consider the total population, not just the skeleton. In particular, the mutation rate scales the spectrum linearly, but this dependence on P naught is a little bit more complex. And we also looked at the fixed size spectrum, where we define tau sub n as the hitting time at which the population first reaches size n. And so finding this spectrum is more relevant for clinical samples where you don't necessarily know the age of the tumor, but you know the tumor size when you get the sample. And so conditioning in the event that this event is finite, we can write down a computational expression for the expected spectrum at this hitting time. And I don't expect you to sort of dwell on these formulas for very long, but this, I'll just mention, this also matches up with the prior approximations at the place when P naught equals 0. And so maybe it's easier to look at some figures to see some differences between the fixed size and the fixed time. This is pretty intuitive. If at some fixed P naught, then showing the theory and the simulations graphs that my student made, you can see the deviation between these two at the large frequencies. That is due to the fact that in this fixed time spectrum, you will have many times have more than n cells at time Tn, and that isn't possible at the fixed size spectrum. And of course, you have a point mass at n representing all the clonal mutations in the fixed size spectrum. And I'll just note that as the number of cells goes to infinity, the difference, when n goes to infinity, the difference between these two spectra decreases. And to give you an idea of what is n in a tumor, then typically a tumor reaching the size of 1 centimeter cubed or 1 gram mass is going to have approximately 10 to the 9th cells. So to better understand how changing P naught is going to affect this SFS, let's recall that the expected value of Sj in the large n limit behaves at fixed time Tn behaves like this. And for a fixed P naught between 0 and 1, if we send j to infinity, so remember j is the number of cells the mutation appears in. So we're looking then at the large frequency end of the spectrum. Then in this limit, then we really do get this thing that is the 1/j² law in the prior approximations on the skeleton. On the other hand, if we fix j and send P naught to 1, so that's going to be a tumor with a lot of cell death, then this looks more like a 1/j law. And this matches up actually with the same frequency spectrum of fixed population size models. In particular, for example, the neutral Moran model, where the expected number of mutations in J cells of a sample size, sorry, my notation is wrong here, the sample size should be capital N, also follows this 1/j law. So when P naught equals 0, the SFS of the total population is accurately captured by the SFS of the skeleton, which is why our results at that point matches up with prior approximations. But when P naught is bigger than 0, the SFS still follows the 1 over j squared law at the large frequency end, but it deviates at the small frequency end. And as P naught approaches 1, it transitions to the 1 over j law of the constant Moran model. So here you can see this in the figures. Just check the time. For two different values of n, 10 to the 4th and 10 to the 5th. And here we put P naught to be 0.99. So what you're looking at is the SFS. On the x-axis is the number of cells. And so the SFS is in the solid line, and the red dashed line is the 1 over j squared law, and the green dashed line is the 1 over j law. So you can see this transition between the two power laws. And to kind of get some intuition for this, note that the mutations that occur early on in the evolution of a large tumor are only going to be detected if they occur on the skeleton, which is why mutations at large frequencies follow the 1 over j squared law of the skeleton spectrum. But if you have mutations that occur late or right before sequencing, these mutations are present at small frequencies. So you need to consider both the skeleton cells and the cells whose lineages eventually die out. And when P naught is large, so that's a high cell death, most cells are ones whose lineages eventually die out. And so when it approaches 1, these clones start to behave like a critical branching process with net growth rate 0. And that's why these mutations start to follow the 1 over j law of a constant size population. So you might be wondering, how relevant is this large P naught regime in cancer? And the answer is that there's typically a lot of cell death in tumors. And so there's a couple of papers here I'm citing in recent work where the P naught parameter is being estimated in other ways. And you can see across many different cancers, the P naught is quite high and quite close to 1. Okay, so we also wanted to establish some laws of large numbers for the fixed time and fixed size spectra, so that we can apply the expected spectrum results to understand the evolutionary history of individual tumors, SFS. And in particular, we find that as n goes to infinity, this fixed time spectrum converges in probability to its mean multiplied by x, an exponential and invariable with mean 1. And the fixed size spectrum has the same limit except for the x, which of course captures the variability in the tumor size at time tn. And let's see, here's some figures demonstrating the law of large numbers at fixed time for n equals 100. Let's see, n equals 100 and 1000, and then fixed size for n equals 100,000. And for here, of course, shown converging to a constant for the fixed size and the random variable for the fixed time. Since this variable x in the limit here does not depend on j, if we normalize the fixed size spectrum by the total number of mutations, then it will obey the same law of large numbers as the normalized version of the fixed size spectrum. So that gives us kind of a nice way to relate these two similar quantities. And that gives us some more flexibility with dealing with either clinical or lab type of data. Okay, so these results turn out to be useful for designing a simple metric for inference of those parameters W and P naught that we're interested in. So let's let S1 be the expected number of mutations in one cell. And let's let M1 be the total number of mutations. Then we can calculate for the fixed time spectrum, the S1 over M1 and its limit, which are independent of W, and they are functions of P naught. So then using this, we can define a function phi sub n of P naught. And to be the expression that we've derived for S1 over M1. And so if we know S1 over M1, then we can invert this phi to obtain what we're interested in, which is P naught, which allows us to estimate P naught and W. So this figure kind of demonstrates how this metric S1 over M1 kind of captures the transitions between the 1 over j squared law and the 1 over j law at the small frequency end of the spectrum. So here, the x-axis is P naught, and we're plotting S1 over M1 as a function of P naught. And S1 over M1 converges to 0.5 as P naught goes to 0. In this regime, the SFS of the total population is the SFS of the skeleton, in which case half the mutations are found in one cell. So that's the no death regime. And then on the other hand, if P naught goes to 1, the SFS mirrors the SFS of the constant population Moran model, in which case the expected number of mutations is like 1 over log n to large n. Sorry, the proportion of mutations is 1 over log n for large n. Okay, so this metric does capture this sort of transition between power laws. And so to estimate P naught, to apply it sort of practically, we can sample a subclone of size n from a tumor. So we define a subclone as all currently living descendants of a given common ancestor. And we can calculate, or not calculate, but read off S1, the number of mutations found in one cell of the sample, and M1, the number of mutations, total number of mutations in the sample. And then we just define this estimator P hat of n, which is phi n inverse of S1 over M1. And this estimator is consistent, that it accurately recovers the true value of P naught given a sufficiently large sample. So to test out this estimator, we generated some synthetic data from multiple independent subclones of size n with true extinction probability P naught varying here. So here we have n equals 200 in the top row and n equals 1000 in the bottom row, and P naught varies in each column. You're looking at the estimated value of P naught. And so you can compare with the true value. And so we can see that as the size of the subclone increases, in each case, the estimate gets better and sort of the histogram concentrates around the true value. And you might be wondering about this mass at 0. And so that's coming because in the ratio of the expected values, S1 over M1, this ratio takes values in the range 1 minus 1 over n over log n to 1/2. But then in the sampled ratio, this actual S1 over M1 can be anything from 0 to 1. So we kind of have to extend the definition of the inverse of phi. So if S1 over M1 is bigger than a half, then the estimate for P naught defaults to 0, since the expected ratio S1 over M1 is the largest. And then otherwise, then so we basically have to extend this inverse definition on both ends. So analogously, it will default to 1 if it's lower than the theoretical attainable value. Okay, these are some performance metrics for the estimator. So we can see the mean and the median, and the standard deviation for the estimates for n equals 200 and n equals 1,000. This is the same. This is like summary statistics of the data on the previous slide. But you can see that the standard deviation improves as n increases and as p increases. Okay, so that's pretty much all I have, and I wanted to just mention in the end some limitations of this. So in the practical application of this estimator, one of the things we stipulated at the beginning was that you have to extract the entire subclone. And so it's unclear how feasible that is in a real setting. And the other major limitation is difficult right now to accurately observe the small frequency end of the spectrum, which is what is needed to decouple W and P naught. So both of these are real practical limitations. One I think of as being addressable from the theoretical standpoint, the need to extract entire subclones. We can, of course, and we are working on developing this analysis for sampled SFS instead of entire subclone SFS, and I think that's something that's possible. On the other hand, the observation of the small frequency end of the spectrum, that's more of an experimental limitation, and there are single cell sequencing is available now, where you can actually identify the mutations present in individual cells, so that's theoretically possible right now, but it's expensive to do that. But every day it's more things the technology is improving, so it's sort of becoming more and more accessible to get at this small frequency end. Okay, so in conclusion, we derived exact expressions for the fixed time and fixed size of the SFS for neutrally evolving tumor. We see that the mutation rate and the extinction probability P naught decouple at the small frequency end. And as P naught increases from 0 to 1, at this small frequency end, the SFS transitions between 1 over j squared and 1 over j law. And we can, in principle, exploit this transition to estimate P naught and W using only the SFS. So that, I think I'll stop there and see if there are any questions.
Thanks for a very nice talk, Jasmine. Are there any questions in the room or online?
Hi, Jasmine. Thanks. Did one of your slides gave like estimates for P naught from real life tumors? Was that right? And that they were close to 1. So how did those people get their estimates for P naught?
That's a good question. See if I can remember. I think these were done in different ways.
They weren't using your method, or because you seem to be saying that your method was.
Yeah. So I mean, typically, and I'm sorry, I don't remember the specifics of these particular papers, but one of the ways that you see P naught being estimated in real cancer cell lines or tumors is typically to look for net growth rates, and then to do some staining for cell death over time.
Okay.
And that way you can decouple the birth and death rates.
So your method would be able to estimate it just from a snapshot rather than from looking at it over time, I guess. Is that, yeah.
Right, exactly.
Yeah, thanks, Jasmine, for a very nice talk. And please stay on that slide. So, so as far as I understand, there is some transition going on between Kingman coalescence and Bolthausen-Schnittmann, right?
Mm-hmm.
So can you give a bit more details about that? So what do we learn from this, from the genealogy, for the genealogy?
So it's really just driven by the fact that when we look at the large frequency end of the tumor, we're really looking back in time at the mutations that occur early on and on the skeleton, so the cells with infinite lines of descent. So the previous results on the 1/J² law sort of used the approximation that the made these calculations on the skeleton. And when you go to the small frequency end, you're having to take into account the contributions from these lineages that are going to die out. And so at this point, when P naught is large, then you have quite a lot of cell death, and you have almost like a critical branching process. So you end up transitioning from the 1/J² law to the 1/J law, which kind of reflects these constant population size.
Right. I'm just trying to untangle. I mean, to have the genealogical picture plus the mutations all together, and like looking at it not in terms of the mutations, but more rather in terms of the tree and then what we see in the tree. So it's maybe unclear, but I mean, as you say, the side frequency spectrum doesn't give all information, right? It only gives crude information, and I'm just wondering what is the full information that you can expect from that.
Oh, you mean like if we were to consider the properties of the entire tree instead and try to use those information to try to infer? Yeah, that's something we're working on right now. Yeah, I don't have a ton of insights right now or results to share right now on that, but that's something that, is sort of clear from all of this, that the side frequency spectrum is not really the best tool to get at, inferring these evolutionary histories.
I just wanted to follow up on Pascal's comment about the genealogy, so that you do see 1/J² from Bolthausen-Schnittmann, but you will also see 1/J² just taking a sample from a supercritical branching process with no multiple mergers at all. So you can't automatically conclude the Bolthausen-Schnittmann from the tail of the side frequency spectrum. So here, I think she's just seeing a supercritical branching process, and that gives the 1/J².
One question, Jasmine. So my understanding of hearing some other talks about colorectal cancer is that the so-called Big Bang hypothesis is coming to be the more accepted model, but that seems inconsistent with what you were saying about P naught, your interpretation of P naught. Am I confusing ideas, or is that?
Can you elaborate on that?
Well, you said that when P naught is large, as is the case for colorectal cancer, that suggests that.
So.
Whereas my understanding is that a lot of the clones appear very early in colorectal cancer.
Yeah, I guess I kind of view these as different. Different issues or mechanisms. So as far as I understand, the Big Bang hypothesis kind of refers to early on a lot of genomic instability and mutations kind of occurring early on in the process. And we could, so this model, so the way I would interpret that in terms of the model would be something like a transition in the mutation rate in time. So that's not something that we have incorporated into this model. So I don't think that we can say much about the Big Bang hypothesis using this. I mean, I mean, it's an interesting idea to maybe add that in as a feature to the model and see how that would change the site frequency spectrum.
All right. Thanks very much for a nice talk.
Thank you.